A mouse swims out of a rainforest stream. A fish stares back with a swollen, blob-shaped head. High above the ground, a salamander clings to a tree. Then, on a wet leaf, researchers find a frog so unfamiliar that it does not yet have a scientific name. How can animals still be hidden in the Amazon, one of the most famous ecosystems on Earth? The short answer is that knowing where a forest is does not mean knowing everything inside it. Much of the Amazon has been sampled unevenly. Many species are active only at certain hours or seasons, and some look almost identical until their calls or DNA expose the difference. These animals are not suddenly appearing. They were there all along. What changes is our ability to notice them, compare them, and prove what they are. A striking example came from the Alto Mayo landscape in northern Peru. During a 38-day survey, researchers recorded more than 2,000 species. Their report identified at least 27 animals judged to be new to science, including 4 mammals, 8 fish, 3 amphibians, 10 butterflies, and 2 beetles. Another 48 plants and animals were listed as potentially new and in need of further investigation. That difference matters. A potentially new species is a candidate. It may look unusual, live in an unexpected place, or carry DNA unlike known relatives. But scientists still need to rule out other possibilities. It could be a juvenile, an unusual color form, a hybrid, or a known species whose appearance varies across its range. Even the 27 Alto Mayo animals identified as new to science did not instantly become 27 finished entries in the official catalog of life. Finding an organism in the field is the beginning of a taxonomic investigation, not the end. And the first obstacle is simply getting to the right place. On a map, the Amazon looks like one enormous green region. On the ground, it is a maze of rivers, flooded forests, upland forests, swamps, bamboo thickets, cloud forests, isolated ridges, and streams that may be reachable only by boat and days of walking. Research follows logistics. Scientists tend to sample near roads, navigable rivers, towns, and research stations. A major analysis of thousands of ecological survey sites in the Brazilian Amazon found enormous gaps, especially in upland forest. In other words, the places represented in scientific databases are not a perfect cross-section of the forest. They are often the places humans can reach repeatedly. Now shrink your perspective. To a tiny frog, a river can be a wall. To a small fish, a waterfall can separate populations. To an insect that depends on one plant or one soil type, a change in vegetation can mark the edge of its entire world. The Amazon is not just large, it is divided into countless biological neighborhoods. Populations separated by rivers, ridges, or unsuitable habitat can gradually follow different evolutionary paths. Over long periods, they may develop different colors, calls, behaviors, or genes. Some species can occupy a surprisingly small range, perhaps one drainage system or one patch of a particular forest type. That makes them easy to miss. A survey can pass only a few kilometers away and never enter the habitat where they live. Time creates another hiding place. Walk through the forest at noon, and you sample the noon forest. Return after dark in heavy rain, and it becomes a different biological world. Frogs begin calling, moths arrive at lights, spiders reflect headlamps from the leaf litter. Small mammals move through stream banks and low vegetation. Some amphibians call for limited periods during the rainy season. Some insects emerge as adults for only a short window. A species might be silent, underground, high in the canopy, or present only as an egg or larva during the exact week researchers visit. That is why night surveys can resemble detective work. The scientist may hear the animal before seeing it. A faint chirp comes from a wall of leaves. The sound stops whenever someone approaches. Then a headlamp catches a tiny throat inflating on a plant stem. For frogs, that call can be as important as the animal's face. Two frogs may look nearly identical to us, yet use different mating calls. Differences in rhythm, note length, or frequency can prevent them from recognizing each other as mates. What humans had treated as one widespread species may actually be several hidden or cryptic species. DNA has made those divisions easier to detect, but it is not a magic machine that scans an animal and prints a name. Researchers compare genetic sequences from multiple individuals and populations. They look for lineages that have remained distinct. Then they combine that evidence with anatomy, coloration, calls, behavior, geography, and natural history. The strongest case usually comes when several clues agree. This is exactly why two tiny Amazonian poison frogs, formally described in 2025, are so useful. Both came from poorly sampled forests in Brazil's Juruá River Basin, but neither was named from appearance alone. Ranitomeya aquamarina has metallic blue-green stripes and was studied using body measurements, morphology, advertisement calls, genetic data, and observations of its habitat and breeding behavior. Soon afterward, researchers formally described Ranitomeya aetherea, a frog roughly the length of a thumbnail. Its back carries pale blue stripes, while its legs appear metallic copper. Calling males were found perched on leaves and in plant sheaths above the forest floor. Tadpoles developed inside tiny pools of water held by plants. A frog that small, living around particular water-holding plants in a remote basin, is not easy to encounter by accident. To establish Ranitomeya aetherea as a species, researchers compared its body, color pattern, tadpoles, calls, and DNA with known relatives. They designated a holotype, the physical specimen that anchors the name, placed specimens in scientific collections, and published a detailed description that other experts could examine. That is the line between an exciting candidate and a formally described species. Formal description can take years because the new animal must be compared with species described across centuries. Museum specimens need to be examined. Old papers may need to be translated. DNA must be analyzed. Calls must be measured. Specialists may discover that the same animal was collected decades earlier but placed in a drawer under the wrong name. And there are far more mystery specimens than taxonomists available to study them. There is also a human problem hidden inside the word discovery. An animal may be new to academic science without being new to the people who live beside it. Indigenous communities and other local residents may know where an animal lives, when it appears, what it eats, or how it behaves, long before a visiting researcher arrives. The Alto Mayo expedition included local experts from Oahuan communities. That knowledge helps teams navigate the landscape, recognize seasonal patterns, and search habitats that an outsider might overlook. So the most accurate discovery story is often not a lone scientist entering an untouched wilderness. It is a collaboration between taxonomists, field biologists, museum collections, genetic laboratories, and people with generations of knowledge tied to the land. Finding hidden species matters because conservation decisions depend on names and ranges. Imagine one frog believed to live across a huge section of the Amazon. It appears secure. Then calls and DNA reveal that it is actually 5 species, each confined to a much smaller area. The total number of frogs has not changed, but the conservation picture has. A single road, fire, mine, or polluted stream could now threaten an entire species. Without a formal identity, an animal can be almost invisible to environmental assessments. Once it has a name, researchers can map it, monitor it, evaluate threats, and argue specifically for the habitat it needs. This creates an unsettling race. The same remoteness that hides species from science can protect them for a while, But roads, deforestation, fires, and climate change can reach poorly studied habitats before taxonomists do. The real mystery, then, is not how a bright blue frog or an amphibious mouse could remain invisible in a famous rainforest. The mystery disappears once we stop imagining the Amazon as one well-explored place. It is millions of streams, trees, ridges, seasons, and nighttime worlds, each filtering life in a different way. Scientists are not reaching the end of a finished catalog. They are opening tiny windows into a forest whose map is still full of biological blank spaces. And sometimes, inside one of those blank spaces, a leaf begins to call.